We are starting with slide 24 of the chapter 1 lecture notes, and reminding you that you need to know the levels of classification from domain, which is the most inclusive, includes the greatest number of organisms, all the way down to the species group, which is least inclusive. So it's domain, supergroup, kingdom, phylum, class, order, family, genus, and species. For example, humans are in the domain Eukarya, which means that the cells that make up a human being, the trillions of cells that are found in the human body, all have a nucleus. Well, for the most part, the red blood cells lose their nucleus when they become mature, but we're just saying in general, the cells have a nucleus at some point. So humans are in the domain Eukarya. The supergroup is the Opisthokonta, which includes the animals and the fungi, and then some of the protozoans. The kingdom is Animalia, which includes the animals. The phylum is Chordata, which just means that humans have a notochord. And most of the members of the phylum Chordata are vertebrates. So we'll just keep it simple. Most of them have a backbone. The class for humans is Mammalia, so humans are mammals. They have hair and mammary glands. The order is Primates, and these will be the primates have opposable thumbs and include the apes. And then the family is Hominidae, the genus is Homo, and the species is Homo sapiens. You can see the different classification groups for corn. I want you to focus down on the species, because the species is also showing you the scientific name for the human and the corn. The scientific name for humans is Homo sapiens, or you can put H. sapiens. For corn, it's Zea mays, or you can just put Z. mays. But it's the genus name capitalized and the species name lowercase together. So the species name and the scientific name are binomial. They're two-part names. And here it is, Homo sapiens down here at the bottom. So we've learned two things when going through the classification groups, is to know the classification groups from the domain to the species, and then also know how to write the scientific name of an organism. And you would also, by the way, take the genus and species name, and you would put it in italics, or you would underline both names. All right, the three domains are Archaea, bacteria, and Eukarya. The domain Archaea contains unicellular prokaryotes. That means they're single-celled organisms made of one cell only. Their cells are prokaryotic, meaning they don't have a nucleus, and they're small compared to eukaryotic cells. They, in the Archaea, live in extreme environments, probably similar to the primitive Earth. Very hot, very salty sometimes, very acidic. And just remember, prokaryotes lack a membrane-bound nucleus. The second domain of living things that are prokaryotes is the domain bacteria, which is probably more familiar to you. Bacteria are also unicellular prokaryotes, but they're different enough from the Archaea that they cannot be in the same group. All right, in the domain Eukarya, there are some that are unicellular. Most are multicellular. And eukaryotic cells contain a membrane-bound nucleus. So that's the difference in the cells that form organisms within the domain Eukarya versus the other two domains. Here's a picture of Sulfolobus, which is in the domain Archaea. And it just says that in the domain archaea, the cells are prokaryotic. They can have various shapes. They are adapted to extreme environments where most living things today would not survive. Very hot, very acidic, or very salty environments that contain toxic gases, that kind of thing. They absorb or chemosynthesize food, and they have unique chemical characteristics in comparison to the domain bacteria. These bacteria within the domain bacteria are Escherichia coli, or E. coli. The domain bacteria includes prokaryotic cells of various shapes. They're adapted to all environments, even inside and on the human body. They absorb or chemosynthesize food, but also we have some photosynthetic bacteria called cyanobacteria. And they also have unique chemical characteristics that make them too different to be put in the same group as, or same domain as the archaea. All right, then we have the 4 kingdoms within the domain Eukarya. The kingdom Protista includes the algae, protozoans, slime molds, and water molds. They can be complex single-celled organisms, or they can grow as filaments, colonies, or they can be multicellular, like multicellular algae. They absorb, photosynthesize, or ingest food. They have different ways of obtaining food. What you're seeing here is a picture of a paramecium, which is a single-celled protozoan. Then you have the kingdom fungi, which includes molds, mushrooms, yeast, and ringworms. Most of these are multicellular. There are a few that are unicellular, like yeast. You have the kingdom plantae, which include mosses, ferns, conifers, and flowering plants. Plants are multicellular, and their cells are capable of photosynthesis. And you have the kingdom Animalia, which includes sponges, worms, insects, fishes, frogs, turtles, birds, and mammals. All of the members of the kingdom Animalia are multicellular, and they ingest their food. You see a picture of a red fox here. The domains archaea and bacteria, we don't really focus on the kingdoms within those domains, but we do have 4 kingdoms within the domain Eukarya: Protista, fungi, plantae, and Animalia. The supergroups are, you won't have to worry about learning these until you get to Bio 112, General Biology Part 2, next semester. But because when we learn about the kingdom Protista, we learn them within their supergroups. We divide them up into supergroups. But again, I will tell you, so like the Opisthokonta includes the kingdom fungi, the kingdom Animalia, and then the protozoans called choanoflagellates. And then the plants are within the Archaeoplastida supergroup. That includes red and green algae and land plants. Now, for scientific names are universal. In other words, you can use those names in when you're speaking in any language or communicating in any language, and that they would be recognized as the specific organism that they represent. There wouldn't be any confusion. They are Latin-based. Scientific names are written using binomial nomenclature. That means a 2-part name. The first word is the genus. It's always capitalized. Second word is the species or specific epithet, and that's written in lowercase. You italicize both words, or if you're handwriting it, you underline them. So again, we see the scientific name for humans is Homo sapiens, capital H, little s, and both words are italicized. And corn is Zea mays, capital Z, little m, both words are in italics. Now, In this next section, the scientific method, I'm just going to introduce it because we will use lab to learn the scientific method. You do need to know the scientific method is used to solve problems, but it cannot be used to solve every problem. There are certain problems that it lends itself to helping, to solve. And then some problems like, I don't know, which color is the prettiest color? That's really hard to use the scientific method, for opinion type problems. Now you could do a poll and find out how many people like green and how many people like red and how many people like blue the best, but it still isn't completely a problem you can solve with the scientific method, finding out which color is the prettiest, because pretty is different to different people. It is subjective. Also, the scientific method really is not very useful for solving problems of a spiritual nature. So is there a God? Paranormal type questions. But for a lot of problems involving the natural world, the scientific method is very useful, and it has a specific sequence of steps: observation, hypothesis, predictions and experiments, data collection with statistical analysis, results and conclusion. This is a diagram that goes through the scientific method in a lot more detail than what we're going to in class, but I do want you to know what the definitions are of some of these terms. So a hypothesis is defined as a tentative explanation for the question or the problem that you're trying to solve. So a tentative explanation for a problem or a question, that's a hypothesis. So it is a statement that you can test, it's testable, and it's developed through the process known as inductive reasoning. An experiment is a series of procedures designed to test the hypothesis, and you use deductive reasoning to make a prediction, to predict the expected outcome of the experiment. You do need to know the difference between the independent variable in an experiment and the dependent variable. The factor being tested is the independent variable, and then the condition that we're measuring is the dependent variable. So let's look at, you also have two different groups in an experiment. A test group is exposed to the experimental variable, which is your independent variable. And the control group goes through all aspects of the experiment, but is not exposed to the experimental variable. So looking at this experiment, variation in blood cholesterol levels, over several weeks. I don't really understand what this experiment is. So, okay, so, all right, let me just tell you one. So in lab, we're going to do an experiment where we expose pill bugs or roly-polies to sand and cornstarch. So they're going to be able to choose to either they'll be in a choice chamber. You're going to do this as a virtual lab, and then you're going to do a real lab with pill bugs. But the virtual lab exposes the pill bugs to a choice chamber. So you put 10 bugs in the choice chamber, and you count how many are on the side with sand and how many are on the side with cornstarch every 2 minutes for 20 minutes. So what we're trying to do is see if they prefer sand or cornstarch. So the independent variable is the sand versus the cornstarch, or you could say here that it's the cornstarch. The dependent variable, which we're measuring, are number of pill bugs choosing sand versus number of pill bugs choosing cornstarch. So number of pill bugs is the dependent variable. So what you measure is the dependent variable. What you are actually testing is the independent variable or experimental variable. Then you have control variables, which are variables that are kept constant. We'll keep the temperature constant, for example. The amount of light will be constant, that kind of thing. And then we're just looking here at the x-axis is going to show your